Welcome back, everyone, to the Allen School's Molecular Programming Seminar Series. And joining us today is Professor David Doty from UC Davis's Computer Science Department. Dave's one of the leading figures proving and also demonstrating how chemistry, biology, and physics can be used to perform computation. And I'd say he's a central figure in molecular programming in a really strong sense, since he was responsible for many of the important theoretical results across nearly every major computational model that we look at in the field. Not being satisfied with only proving wonderful theorems, he's also responsible for one of the most impressive experimental demonstrations, which we'll hear about today. And on a personal note, I'll say Dave's also one of the best pizza chefs I've ever met. Welcome, Dave.
Okay, thank you very much, Chris, and thanks for inviting me. Really excited to talk about this. So this is a project that took a very long time. It took a very, very long time, but I started it when I was a postdoc back in the same lab that Chris Patrick used to be a postdoc in, along with Damien Woods, who is now a professor at Maynooth University in Ireland, but he was a postdoc back then, and we were both working for Eric Winfrey doing this project. And we had a bunch of other people who contributed very significantly as well. So what do we do? We're interested in building things, right? That's what we focus on. We build things. Now, this is an old technology. People have been building things for a long time by hand, or maybe they use tools. And this works great for things that are approximately the size of a person, give or take. One idea that became very prominent in the Industrial Revolution was building tools that themselves build things, right? So maybe rather than building a car by hand, you actually specify a computer program that tells a bunch of robots how to build a car. And an early example of this is the Jacquard loom. So if you heard about old computer programs use punch cards, like that technology came from this loom that would weave patterns. And this is actually a picture of Jacquard woven into a loom that looks like him. So this is a very common approach as well. So what we're interested in is a variant on these. It doesn't quite map onto these, which is things that build themselves, right? So rather than a tool building a different object, we're looking at something we call self-assembly, like objects that construct themselves. So this is useful if you have some idea that you want to construct something in a small place, maybe, or kind of a biological place where our hands or our tools simply can't reach, without disrupting it. So what would such an idea look like? What would it mean to have an object that builds itself? Well, if you think about like maybe these Lego bricks here, like how do you form this? Well, you put them together, but how would they self-assemble? Somehow this red Lego piece would have to know, somehow I want to stick below blue and yellow, but above this blue and this green right here, so that if you just toss them all up in the air and just let them collide with each other, like these kids are doing here, without any guidance from you, they'd automatically assemble into some complex structure. So that's the idea of self-assembly. And why do we want to do it? Well, because this is one of our cheaper and more reliable options if these are very small molecules that we can't manipulate by hand so easily. So, so I'm going to talk about this self-assembly project. And like any respectable computer scientist, if I have a complex system that I want to think about, I don't try to think about all of it at once. I want to break it into different layers of abstraction, where when I'm sitting at a certain layer of abstraction, I'm not thinking about how the one below me is implemented. I'm just trusting that somebody, actually in this case, Damien and I did all of these layers, but when you're working at one layer of abstraction, you assume the one below you has been sort of implemented, right? So you write a high-level programming language in Python. You don't worry about how to turn it into assembly code instructions. And if you write assembly code instructions, you don't worry about what their binary representation is in the processor. And if you were even thinking about that, you don't worry about how those binary bits translate into voltages that go into transistors, right? Each of these needs to be worried about at some level, but when you're at one layer of abstraction, you don't worry about the ones beneath you. So eventually we're going to get to the full goal of this project, which was to make DNA strands that implement, something, But what are they doing? They're implementing computation on bits. So we're going to start at the highest layer of abstraction, which is just Boolean circuits that do computation on bits. So, okay, so let's start simple. There's some bits, right? There's some bits. And I don't like them too much, right? I'm not a fan of these bits. I like better to have them kind of sorted. So the ones are on top and the zeros are on bottom. So how can we kind of do this transformation? Well, we might imagine if we see a 0 and a 1 out of order, we can swap them, right? Or if we see a 1 and a 0 in the proper order, we keep them in order. And, you know, 2 1s, there's only one order they can have, so you'd leave them alone. And if you kept comparing bits like this, well, you'd never be able to get the 1s down here up here. So we can imagine kind of shifting the reading frame and do the same thing. If a 1 and 0 are out of order, swap them, otherwise just copy. And if we just keep doing this sort of computation, eventually the bits end up sorted, right? So now sorting bits, you know, might seem like kind of a boring task. If you know anything about this theoretical model called sorting networks, you know, typically the things you want to sort would be like numbers or strings, like list of names or something. But it turns out if you have a sorting network, one way to prove it works on any list of objects you might want to sort is to prove that it works for all sequences of bits. And if it works for those, it works for anything else. So even the idea of sorting bits, just shove the 1s to one side, is actually a sort of a non-trivial thing to do. So, okay, so there's a simple kind of computation to think about on bits. Here's another one. I'd like to know whether I have an odd or an even number of bits. So maybe I'll try to get the 1s to this middle part here, sort of to count them. So if I have 1s above that line, I'll move them down. But if I have a 1 below that line, I'll move it up. So what I'm demonstrating here is maybe we want to do different computations depending how high we are, you know, along the bits here. And I can only fit, you know, 1 bit at a time in here. So if I have 2 1s meet, they'll cancel, they'll both become a 0. And so this remaining one will kind of eventually make its way here, and then it just gets copied. And so we have basically an odd number of 1s. So if they cancel in pairs, you'll have 1 1 left over and have this stripe of 1s. But if you had an even number of 1s, you know, the middle 2 cancel, and the other 2 kind of make their way into the middle, and they eventually cancel. And now you do not have that sort of middle row of 1s. So this is some sense in which we can do a computation on these bits and tell, was there an even or an odd number of 1s by the presence or absence of a streak of 1s right here. So what I'm getting at with these examples is I want to just kind of get you thinking about a model of computation we're going to define a bit more formally now. And the model is a circuit model, Boolean circuits. And the fundamental sort of piece of this model is something we call a gate. And all a gate is a function that has 2 bits as input and 2 bits of output, right? So kind of like Boolean gates you've heard of, but often those just have 1 output bit. So the idea is you have 4 possible pairs of input bits. And then for each of the 2 outputs, any bits that you can think of to write down here, that makes a legitimate gate. So maybe the top one might be the XOR function and the bottom one might be the AND function. So that's an appropriate sequence of 8 bits to put there. So that a gate can just be any way of putting bits based on the input. Okay, we have a gate. And it occupies a certain kind of height or row. This one's in row 3, and it connects to gates at the adjacent rows. So the output of 3 goes to a gate at row 2 and another one at row 4. And all the rows put together, there's kind of 7 rows here, make a single layer, as we call it. And as I said, you can have different gates, meaning different functions being computed in different rows. So that's a feature of our model here. I didn't show an example of this yet, but we allow randomization. So maybe what you want to do is flip coins to decide what to do. That's allowed in our model. So we formalize that by, you might have 2 different gates at a single row, and one of them gets picked randomly. It might be half probability of each. You might set the probabilities to be different than a half. So that's something you can do in the model. So the high level overview is you, the programmer, program in this model by specifying a layer, meaning what gates do you want to be computing in each row. And then the user is going to kind of give you some bits that come in from the left, right? So these are the input bits. And then you get to pick these gates to be whatever you want. But now, whatever you picked them to be, now they repeat as you go to the right, right? So this 3 can be a different function than this 5, but it's the same function being computed as this 3, and it's the same as being computed by this 3. They might be taking different bits as input, but they're computing the same function on them. So that's the model. So go ahead and ask any questions if you're not clear on what that's doing, because this is important for what we're doing. So, okay, so let's look at some more examples. So here's like the simplest sort of computation you could possibly do in this model. We just call it copy. Just whatever is your top input bit, that's your top output. Your bottom input bit becomes your bottom output. And I'm going to start drawing the circuits a little differently now. I don't want to have all this clutter of the various gates and their functions. So what I'm going to do is just draw a little gold dot wherever there's a 1, and the absence of a gold dot is a 0. And we're kind of ignoring the information about, where are the gates, but, implicitly they're under here. And we can just visualize how ones move through the circuit as computation flows. So this is what copy would do. The ones would just kind of get copied to the right. And we saw sorting already. So what this would do is if you start with some ones, say here on the bottom, they would sort of move up as you go to the right until eventually they get to the top. And then it would they'd stay at the top. And the computation kind of goes forever to the right. So now these use basically the same computation in each row, but we don't have to. We can put different gates in each row. Parity is an example of that, where it looks different on the, above the middle versus below the middle. And this, will move ones into the middle, canceling them in pairs. So the stripe of ones or not tells you the parity of the number of ones you had to begin with. So these are very simple computations I'm showing. So you might think, okay, great, you can do simple computations in this model, but these aren't super sophisticated. So here's something that's maybe a little more challenging, right? So here is a binary string, and I'm going to interpret it as encoding a natural number in binary. So this is the binary expansion of some number, and I want to know, is it a multiple of 3? So can anybody tell me, is that a multiple of 3? Right. So unlike parity or sorting, it's not so fast to just glance at it and immediately tell what the answer is, but it turns out it is. That's the number 27 in binary, which is a multiple of 3. And what it does is it makes this pair of stripes on any multiple of 3, it turns out, and it'll make a different pattern if you don't have a multiple of 3. So how about if we flip the most significant bit to a 1? Right? Is that a multiple of 3 or not? And again, not so obvious, right? So turns out that one's not. That one is 2 more than a multiple of 3. It's 59. And it makes this different pattern. And in fact, this pattern emerges on every multiple of 3. This pattern emerges on every non-multiple of 3. So a little more sophisticated computation than just, parity. As I mentioned, we can do randomized computations. Here's a very simple one, a little pointless to do it, but it's something you can do, which is instead of sorting, you could flip a coin to decide whether to sort or just to copy. We call this lazy sorting. So sort of eventually you get enough of the right coin flips, the ones all move to the top, but it just takes longer than if you were doing the sort gate on every single layer. So there's something else you can do. Okay, so there's other sort of deterministic circuits we can imagine building. Another kind of non-trivial computation to do is palindrome. I've got some bit string as input. I'm kind of writing it vertically here, even though we normally write them horizontally. And a palindrome is a string that equals its own reverse. It reads the same forward and backward, like this band's name here. So this one is a palindrome because it's 101101. This is not a palindrome. And it turns out there's a circuit you can design that on any palindrome will just make all zeros eventually. And on any non-palindrome will make this pattern. So now, one thing to notice is each layer takes 6 bits as input, and then it produces 6 bits as output that goes into the next layer. Right, so you take those 6 bits, you transform them, or maybe not. This layer on input all zeros produces all zeros, so it repeats forever. Same thing here. These do something different. These, like on input, 1 and then 5 zeros produces a 1 and then some more zeros and then a 1 down here. So they somehow change the bits or not, but there's only 2 to the 6 or 64 different strings of length 6. So if you have a deterministic circuit, at some point it has to repeat, right? So before you get to 64, by the time you get to 64 strings, you go any longer, you have to repeat. So you might ask, well, can we make a circuit that would actually iterate through all 64 different strings of length 6? And this is something we tried pretty hard to do. We couldn't quite figure out how to do it. But we did find by computer search, okay, there's a circuit that can get through 63 of the 64 different strings before it finally repeats the string it started with. So this sort of counts as high as possible, but we couldn't actually figure out how to make it count up to 64. So this particular circuit, if you give it the one string I left out here, that's just a fixed point, and it just iterates that forever. And another sort of much more sophisticated computation they can do is that there's a different model, kind of related, called cellular automata. If you don't know what it is, I'm not going to get into the details, but it's kind of a similar model to this, where you have these binary states, and then based on sort of 3 neighbors, they update. And there's this one called rule 110, and if you start it with this bit string, it kind of looks like this as it gets copied. And Damien figured out how to simulate this. Now, each cell here, we only get to do a few of them here, but if you started it with bits like this, it would just make kind of these repeating triangles. So okay, so why is that interesting? These triangles aren't intrinsically interesting, but this rule is very interesting, because if we had more bits than just these 6, this rule, it's known, and Damien actually was one of the people to help prove this, it's known that this thing can efficiently simulate any algorithm whatsoever. So you write a Python program, you can turn it into a starting sort of bit string to give to rule 110, meaning a starting bit string you can give to our model through this simulation, and that can in turn simulate the Python program. So there's a sense in which this is as powerful as anything could be, given the kind of the memory constraints we have. So there's some deterministic computations we can do. We have these other kind of randomized computations. The lazy parity is like parity, but flips a coin to decide whether to copy. Takes a little longer. Random walking bit just does a random walk top to bottom forever. This one makes these kind of diamonds. It's mostly 0, but once in a while you have a random event. You generate a couple bits at the middle, and they go to the edge and come back and cancel. So it just makes diamonds forever. So most of these aren't super interesting. This one I find to be a little more interesting. It's called Faircoin. So this executes this kind of old famous algorithm that John von Neumann came up with. And the idea is, suppose I have a biased coin and I want to make a fair decision with it. I want to decide, are we going to go where I like for lunch or where you like for lunch today? We want to make a fair decision, but we don't have a fair coin. We have a biased coin. It might, and worse yet, we don't even know the bias. Maybe it's 90% heads, maybe it's 5% heads. We don't even know. Can we use this coin to make a fair 50-50 decision? Do we go to my restaurant or yours? So von Neumann's idea was do the following. Flip the coin twice, and if you get the same result both times, heads, heads, or tails, tails, just throw it away. Say, all right, I'm going to repeat. And if you get a different result, you get heads, tails, or tails, heads, those are your 2 options that you report as being, go with my choice or go with your choice. And no matter the probability, heads, tails has the exact same probability as tails, heads. So they have exactly equal probability. So this basically simulates that. It's tossing a biased coin with these randomized gates in the middle, testing, twice, testing to see, did I get the same result? If so, repeat. If so, repeat until you get different results. One result looks like this kind of stripe on the bottom, and the other one just makes all zeros. And this is what you get. Like this and this are each 1/2 likelihood, no matter what are the probabilities of the underlying gates. So there's like an interesting kind of computation you can do with randomization and with some sort of bit processing. So, okay, so there's just a whole bunch of examples of things we can do in this theoretical model that we made up. So, okay, so what's the point of thinking about that theoretical model? Well, the punchline, to jump to the end, is that we implemented every one of these algorithms using self-assembling DNA that implements them, and they all worked with very high reliability. And this is a bit of a gratuitous slide. You're not really supposed to understand what you're looking at right here. All you're supposed to understand is it's really small. Each of these little dots you see is something called a streptavidin protein. But just to show you, that this is the model we were able to implement with computing self-assembling DNA. So I want to show you how we do that now. But this is the lowest layer of abstraction. We're not going to go that low yet. We're just going to go down to the next lowest layer of abstraction. So Boolean circuits are like this fixed object, and then you imagine you feed in some input bits, and the computation kind of flows through the circuit, but the circuit just sort of sits there while the information propagates through it. So tiles are, again, just a sort of formal mathematical model. of squares that float around and they stick to each other, and they go from not being together to self-assembling some structure. So we're going to go from these static circuits that sit there to these tiles that self-assemble so as to simulate the circuit. So how do we do that? Well, we have the gate, right? The gate is the fundamental object in our Boolean circuit model. It has a truth table with 4 rows that tell you, given 2 input bits, what are the 2 output bits? We turn each row of the truth table into a tile. A tile is just a square with a label on each side that we call a glue, right? And here the glues are just these like bits, and each row is one of these tiles, and the 2 input glues, the 2 input bits are represented by the glues on the left side here, and the 2 output bits are represented by the glues on the right side here. So this is like just the transformations. You turn this into these 4 tiles with these bits. Okay, so what do the tiles do? Well, the idea is that tiles that get next to each other and they have matching glues, they want to stick, and if they have mismatching glues, they don't want to stick. So let's get into a little more detail about that rule there. So what's going on? You have this circuit. So how is it that tiles self-assemble so as to carry out the computation of this circuit? Well, the bits correspond to glues. Let's imagine we have some glues just hanging there in space, right? Again, we're in an abstract model here. I've just stuck glues. I don't know what this thing is, but tiles can stick to it. So I've got my 6 bits encoded as these 6 glues, and tiles start sticking. And what we actually have that I didn't really tell you is that each tile sort of has a type that depends on how high it is in the circuit. So I'm sort of labeling this is type 1, 2, 3, 4, and so on here. I'm leaving off the glues for now. We're going to zoom in later and see how do the glues actually play into this. I just kind of want to show you what structure do the tiles form. They kind of bind like this, but then there's a special tile that its bottom half is here and its top half is here. So I have to draw this on a 2D screen, but what I'm depicting actually is not a 2D thing. It's a tube, right? So basically these tiles have a bit of a curvature, and they're actual, they're mostly 2D, but they roll up into this tube. But when I draw them on the screen, I'm going to draw them like this, but you have to understand that what's immediately above this line is what's right here. So this is the structure these tiles are forming as they self-assemble. And again, I'm not drawing the glues yet, so now let's draw some of the glues. So we have glues, they represent bits. They also represent the row. So a 0 glue in row 2 is different than a 0 glue in row 4, right? So I didn't tell you that, but that's one of the aspects. And then now what do we have? Well, remember, for each gate, we have 4 tiles that represent it. So there's a gate, and we want to know what tile is going to go here. So there's 4 tiles that could match this, corresponding to the 4 pairs of input bits we could have. But one of those, or 2 of those tiles mismatch one of the glues. This one mismatches with a 1 on top. This one mismatches with a 0 on bottom. One of the tiles mismatches both of the glues. Only one of those tiles matches both glues. And we're going to say in this model, that's the only one that binds strongly enough to go. We call this cooperative binding. Both glues have to match for that binding strength to be enough for the tile to stick. And then so that tile goes there. And if you recall, the tiles also have the output bits over here. So that's the sense in which this tile binding implements the computation of gate 3, because it's the only one that matches the proper input bits. So that's the sense in which tiles can self-assemble so as to compute what this Boolean circuit does. All right, so that's it for the tile layer of abstraction. So finally, okay, these are perfect square tiles, like they're perfect, and they obey this perfect cooperative binding because I want them to, because this is just a mathematical model. I can do whatever I want. But now what we want to do is say, how do you implement that with an actual molecule implementing the tile that does that? So we're going to do it with DNA. So let's dive a bit into structural DNA nanotechnology. So I don't know exactly, the UW audience often is very largely familiar with this, but I don't know if everybody in the audience knows about structural DNA nanotechnology. So I'll start with a quick kind of tutorial on that. So I like to think of this as DNA carpentry, right? So what does a carpenter do? A carpenter uses a biological material called wood that comes from an organism called a tree, and then does very non-biological things with it, like building tables and houses. And we're going to do something similar. We're going to take this biological material, DNA, we're going to build stuff out of it, but we don't much care that it's originally had a biological function. All we care about is it's got some useful structural properties, even more useful than wood, right? So why would we use DNA as a building material? Well, it's because the specificity of the so-called base pairing means that we can program which pieces of DNA are going to stick together. So often, DNA has this 3D structure that we often draw like this, but sometimes we don't care as much about the 3D structure. We just care there's one strand of DNA and there's another strand of DNA, and they have to sort of point opposite directions to bind, and they have complementary, bases between them. So A binding to T and C binding to G. Now, what's not commonly known, but is true, is that if you have two DNA strands and they're not perfectly complementary, but part of them are complementary. So maybe this part of the red strand is complementary to this part of the orange strand, but not the rest. They can bind just on that part and be kind of floppy elsewhere, right? And here's just a drawing of that happening with the DNA drawn as just lines. So what can you do with that idea? Well, one thing you can do is the most successful DNA self-assembly technique called DNA origami. And this isn't the central to what we did, but we did use it. We didn't demonstrate DNA origami, but we used it as a technology in our self-assembly. So let's understand how DNA origami works. So you start with a very long strand called a scaffold strand. It turns out we use a single-stranded virus to do this. And then you say, well, I want to fold this up. I want this part to stick to this part. So how would I make that happen? Well, I could add a new strand, a synthetic strand that's complementary to this part and complementary to this part on its two halves. And so if you mix them together, you might expect it to bind to both and kind of bring them together. So if you mix several of these that are complementary to various regions of this, along with, in water with a bit of salt, then heat it up and cool it down slowly, you would expect them to kind of fold this long strand into the shape dictated by which parts you chose to bring together. And this is kind of a cartoon video showing, what's a plausible pathway by which you could start from this really long strand and a bunch of these short strands, and they would start binding to that long strand to fold it up. And as you can see, when you get done, what do you have? You have a bunch of DNA helices in parallel, but why are they sticking together? They're sticking together because some strands are in one helix, and then they have a crossover to an adjacent helix, and those crossovers are holding it together, right? So the helices are like a bunch of logs being held together into a raft by rope, only the logs and the rope are actually just both DNA. So this idea works extremely well. This is from Paul Rodeman's original paper showing you can make a bunch of shapes out of this. So we're going to use this in our project as part of what we self-assemble. So what's the point of self-assembling DNA in this? What's the point of using DNA for nanotechnology? Well, there's a bunch of things you can do. I mean, some of them biological because we are using DNA, so it's sort of compatible with cellular stuff. But there's a bunch of non-biological reasons to want to build interesting objects out of DNA as well. This was always one of my favorites, which is a DNA origami kind of clamshell shape that starts out closed, but if two proteins are present that happen to be present in a certain kind of cancer are present, they will pop it open and expose some antibodies. So this was a sort of an in vitro experiment demonstrating a potential kind of first steps towards a smart drug. Of course, I also just think it's cool to build sophisticated stuff out of DNA. So here's just some pieces of art that were made using DNA origami in very sophisticated ways. So so I'm going to get back to what we did in a second, but I do just want to mention one other pretty cool application of DNA nanotechnology. This was sort of a personal project of mine. So these are actually DNA origamis, and I really wanted this to work really badly, so I made a whole lot of them, which is why this surface you're seeing, this is a so-called atomic force microscope, is just coated with them because I really wanted to make sure it worked. So if you kind of zoom in, you can see there's a bunch of little rectangles, and each of them has a little pattern kind of written on it. So it's maybe not clear exactly what I'm showing here, but two of the patterns you can see here are word Beth, and then another one was Yim, and that was the first and last name of my girlfriend at the time. And then there was 4 other words that I made to spell this out. So this was my favorite application of DNA nanotechnology. So I sort of showed this to her in Damian Woods' lab with his atomic force microscope, and then we did some quick training and using some strands that Chris helped me prepare in kind of an emergency. He actually did the mixes to make this. She was able to give me a reply. So that was another successful application of DNA nanotechnology. Okay, anyway, a little gratuitous personal project there, but back to the computation that we wanted to do. So as I said, we have this layer of abstraction, these tiles that we draw like squares. We want to implement them with DNA. There's actually many ways to do this. There's various motifs, things called double crossover tiles, and people actually make tiles out of origami, like really big tiles. We're going to use a very elegant motif. It's called a single-stranded tile, and what it is each tile is implemented by one single strand of DNA. And you just logically, kind of mentally section it into 4 binding domains. And the idea is it's just one piece of it's just one strand of DNA, but each piece binds to one of 4 different tiles. So these aren't shaped anything like squares, but the binding graph that they form is the same thing as if you have squares bind together. So this is the motif we're going to use for tiles. So these have been used successfully to kind of make so-called hard-coded structures. So imagine you put enough of these together to make a big rectangle. Okay, you can make that rectangle, but then if you just leave out certain parts of the rectangle so that the rest are connected, you can make different shapes. And this was shown about 9 years ago by Brian Wei and Mingji Dai in Peng Yin's lab. So now what I want to do now is distinguish this sort of self-assembly and DNA origami from what we're trying to do. So what's the key difference? These use so-called unique addressing. So what's in common between these? Well, in DNA origami, each staple just appears once in the structure. So there's a staple kind of 4 over and 2 up, and it's this one doesn't appear anywhere else. And if you put together many origamis, using each as a tile to form another structure, this is the unique origami that goes in this position. Or these tiles, there's one tile that goes here and it doesn't appear anywhere else in the structure. This is so-called unique addressing. So the distinction with this algorithmic self-assembly we're going to do is that our DNA tiles are going to be reused throughout the structure, right? They're only going to have sort of local information available to guide their attachment because they won't know where they are in it in some sense. So, okay, so we're going to use single-stranded tiles, but we're going to reuse them many times throughout the structure. What kind of structure are we going to form? Well, it's not going to be a flat rectangular structure like this. It's actually going to be a tube, right? So for various technical reasons, I can tell you at the end of the talk if people are curious. Why do we make a tube instead of a flat thing? We actually would have preferred to make something flat. But we made a tube because these are kind of easy, and it was known how to do this. And so here's how to kind of visualize it, is that there's many helices in this 16 helix tube, and they're kind of next to each other. And here's sort of a 3D drawing of actually a 20 helix tube. And comparing it to how I was drawing the single-stranded tiles as these little U-shaped straight lines, this is kind of how they would sort of show up in the tube. So this is the kind of structure we're going to form. Okay, so now let's get back to the sort of information processing aspect of this. Each of these square tiles that has 4 bits on each side. Really, the bits are labeled with the row, but I'm kind of leaving that off here. This is implemented by a single-stranded tile that we logically section into these 4 binding domains that themselves are just, DNA sequences. So we have some circuit we want to implement, identified by these tiles that we implement with single-stranded tiles. Now, we don't want them to start binding to each other on their own, right? This is key. We want a so-called barrier to nucleation. We only want them to be able to bind initially to a seed that encodes the input. So we set the conditions of concentration, temperature, such that these are not very favorable to bind to each other because each of them can only stick by one bond. Remember, one bond is weak. But in the presence of a seed, and we're going to make, this is where we use DNA origami, we're going to make our seed out of DNA origami, but the tiles are going to bind to this. Why are they going to bind to this? Because some of those staples in the origami are going to have these single-stranded extensions coming out the end here that encode the bits we want to give as input to the circuit. And with these sitting here, these can bind on 2 glues instead of just 1, and they can begin growing. And so the idea is you do this, you mix everything together, and you heat it, cool it down to about this temperature and hold for, a day or 2. And the tiles hopefully shouldn't bind to each other on their own, but they should start binding to this seed and grow this tube off the end of the seed. So that's the basic idea of the experiment. Now, we want to look at what happens. So what do we want to know? Well, ideally, we want to know in our final structure, what is the sequence of every single, DNA strand, and where did it go? And we can't just like break it up and use DNA sequencing, because we want to know where are the things. So we can't melt it apart and use sequencing to figure it out. So we want to figure out where are the DNA strands in the final structure. But that's a little bit too much information to ask. We don't really have a good cheap mechanism for making that kind of measurement. So here's what we're going to do. We're at least going to figure out where are the ones in the circuit, right? So we take every tile that encodes a 1, and we stick a little molecule on it called a biotin. And biotin's a really small molecule, doesn't affect the growth energetics that much. It does a little. We had to account for that. So why do we put a biotin on there? Because biotin is known to bind very strongly to this much bigger molecule called streptavidin that we can add after we're done with all this. And it's big enough to show up under an atomic force microscope. So we don't get like the full information of like what are all the tiles and where are they, but we at least see which tiles in the final structure represent a 1. That's where these dots show up. So, okay, so that's what we're going to do. Now, we grow the tubes, but atomic force microscope, AFM, is a 2D technology. It just sort of, it's like dragging a record needle back and forth across a flat surface and just measuring how high it is. So we could put the tubes onto this flat surface called mica and see them with an AFM. They look like this, right? But we don't want to see that. We want to flatten them out and then attach streptavidins and see, where are the ones. So what we do is we remove one of the helices, we call the seam, using this other technology called DNA strand displacement. Once that thing's removed, even though this thing still has some curvature, now topologically, it's a rectangle instead of a tube. So when it hits the mica, which kind of pulls it down because they're attracted, it flattens it out into this flat ribbon, and then this is what it looks like. So, and then we're going to attach streptavidin to be able to see dots on here. I'll show you some images in a minute. So, okay, now there's a little bit more that we had to worry about. The DNA sequences, the choice of DNA sequences to implement these glues really matters. Well, remember, what do we want? We want if a tile matches both glues, matches both DNA sequences, both domains attach. But if only one domain matches and not the other, it shouldn't attach. But, DNA sequences have all kinds of binding energies. If you just randomly pick domains, here's the binding energies of a bunch of domains of the length we have, and here's the sum of the binding energies of a bunch of pairs of domains. And you can see like there's a whole lot of binding domains that, sequences of DNA that are just single domains that are much stronger than whole pairs, that consist of 2 of them if you choose the wrong sequences. So what we would like is to get this kind of separation where in the domains we pick, 2 of them put together are significantly stronger than only one of them. And so by doing DNA sequence design, we can kind of get that to happen, right? Because these correspond to this incorrect binding where one domain matches but not the other. We want that to be significantly weaker than when 2 domains match. And so this is how we enforce that, by designing the energy in this way. And there's a bunch of other goals we have as well. And these are, I can answer questions about what these are if people want to know. But there's other constraints we have for the DNA sequences. So, okay, now what you're going to see, just to prep you for the images, AFMs are a nightmare to use. Some days they don't work at all. Some days it takes hours to get it working. When you get it working, for a few hours, you'd like to collect a whole bunch of images. So what we wanted to do is to do many experiments at once, many circuits, many inputs, and image them all at once. But when we mix them, we want to be able to tell which one came from which experiment. So one of the things we do just for imaging is on that DNA origami, which itself starts as a tube and then unrolls into a rectangle, we wrote some digits on there, again, using the biotin sort of streptavidin trick, where some of the staples have a biotin and others don't, and we choose which ones to put on there based on which experiment we're doing. So for instance, 013 meant we're doing the parity circuit and we're giving it this input. So we mix that with other experiments, we know what the proper, sort of growth off the end of this looks like because this little barcode tag that we wrote on there. So you're going to see these digits. That's why they're there. And incidentally, these digits are what I use to do the marriage proposal later. I figured out how to spell words, just by picking the right subsets of digits. So, okay, so here's the full protocol we do. We have a temperature we want to grow at. And it really matters what the temperature is. Too high and nothing will grow. Things just won't stick together. too low and things will grow, but not because of the seed. The tiles will just start sticking to each other. And we don't want that. We have this sweet spot in the middle where the temperature is just right so that tiles don't stick to each other on their own, but they do stick to the seed. This cooperative binding regime where you need two domains to bind. We mix them all together and we heat it up, cool it down to that temperature. And after about an hour, the seed forms, the tiles are floating around. And then we wait for a couple days and this self-assembly happens. And this is the key, magic here. And then after we're done, we do a bunch of kind of stuff to unzip it and look at it on the mic. So we mix together, this origami seed that's bar-coded with the digits to identify the circuit and the input we gave, these adapter strands that encode the input, the tiles that compute the circuit, and all this other stuff, and do this protocol, and then we can look at the results. Okay, so anyway, how well did this work? I gave a bit of a spoiler already, but how well did this work? So, well, pretty well. So here's a bunch of deterministic circuits, our little simulation together with the digits shown right above an AFM image of what happened. So sorting, properly pushed the bits to the top, parity, properly made a stripe or not, depending how many zeros and ones there were. We tested out a lot of the, all the randomized circuits. And these ones, it's not as straightforward to compare simulation with experiment because the randomization means every time you do this, it looks different. But yeah, these look like what you would expect from, certain random walking bit experiments. Here's rule 110, making the triangles as we expect. Like I said, if we had like thousands more bits, we could actually do interesting computation with rule 110, but we only had 6 bits. Here's fair coin, which if you remember should either make this or this with 1/2 probability, no matter what are the underlying gate probabilities. And what do the gate probabilities correspond to? It means some strands implement one gate, some strands implement a different. We just put them both in at various relative concentrations. And so over a range of like relative concentration ratios that goes from 1 to 9 to 9 to 1 to 50-50, we measured, okay, what's the probability of seeing the stripe or not? And it was pretty close to 50%. Ironically, the one that was furthest from 50% was when we had equal gate concentrations, but it looks like it mostly did what von Neumann's algorithm should do. If you remember, I said we designed a circuit that could go through 63 of the 64 bit strings of length 6 before repeating. So here's an image of that. It takes a very long time to actually see it repeat. And I told you that we failed to generate one that would go through all 64 strings. It turns out that's impossible. So Damien's student Tristan Sterin proved this, that actually there is no circuit that can get through all 63. So this 64. So this is the highest you can count in this model. And it's a consequence of some group theory that he knew that we didn't know. So, and that I still don't totally understand, but I kind of wrap my head a bit around it. All right, that parity circuit, we didn't test all inputs for most circuits, but parity has a pretty easy readout. There's either no ones or a long stripe of ones. So we tested it on all 64 inputs and got, the expected output on those. So we did some pretty heavy testing of this whole thing. So what I want to do now is show you, okay, these are all cherry-picked images. Just spoiler alert, these are cherry-picked images. Not everything worked. So what didn't work? So let's look at a representative sample. Here is a representative 12 by 12 micron image that looks like the kind of thing we did. So let's zoom in a bit and see what we see. So here's a bunch of these ribbons, with these little barcodes to help us figure out which experiment was which. And this is for parity. These are all parity. And the answer should be 1, so you should see a bunch of stripes, and you do. But where are some errors? Well, here's a stripe, and then it goes away. So this was what we call an algorithmic error, and we attribute this to a tile that only matched one glue bound anyway, and flipped the bit from 1 to 0 erroneously. So this happens sometimes. Here are some ribbons that don't appear to have a seed attached. We did pretty careful controls. We don't think these were growing on their own. We think these ripped off from a seed as they were landing on mica, but we don't know. Here's a very frustrating error. About half of the seeds in this image don't have much tile growth on them. So this was like our biggest problem that we just never figured out how to solve. But the basic statistics are about 60% of our seeds did have significant growth into a tube, and these error rates were something like 0.03% per tile attachment. So about 1,400 out of 4.5 million tile attachments. So, okay. So, all right, I'm almost done here. Just give me 2 more minutes. So, all right, so what do we learn from doing this? Well, we made a smallish library of molecules that you can reprogram to self-assemble reliably into many complex patterns by processing information, right? So what that sets us apart from other kinds of self-assembly? Well, periodic self-assembly just means you make a standard kind of crystal that just repeats in 1 or 2 or 3 dimensions forever. So we get more algorithmic control than that. But we use a lot fewer tile types than the so-called uniquely addressed self-assembly that uses a single monomer type for each point in what you're making. And compared to previous algorithmic self-assembly work, well, we just did much more. We made many more algorithms of greater complexity because we were able to use hundreds of tile types compared to the about 22 that they were limited to by previous sort of tile motifs. So this was the first one doing algorithmic self-assembly with single-stranded tiles that let us use a lot more tile types. Okay, so what would be good to do next? Well, here's something I don't like about what we did. We drew really interesting patterns, these algorithmic patterns, but we drew it on a really boring shape, just an infinite rectangle that grows to the right forever. Can we run algorithms with the molecules that will cause them to grow into interesting shapes? So here's a theorem that I learned about in grad school that really inspired me actually to kind of get into this field, which is there's a single set of tile types in that middle layer of abstraction here that can make any finite shape you want just by giving it the right seed, encoding the shape kind of in binary. So you've got a set of tile types, and you use that exact same set, but by putting the right seed into one of them, they'll self-assemble into maybe smiley faces. And by putting a different seed in, will cause these exact same tiles to self-assemble like a dolphin. So these tiles are universally programmable for building any shape. So I don't see any fundamental physical reason this can't be done with real molecules. It's just an engineering challenge to scale it up to that. So I'd like to see something like this happen in my lifetime. That'd be awesome. So thank you very much.
All right, thanks very much, Dave. Are there any questions? I'm going to start with one. Dave, can you remind me, so from a practical standpoint, it's clear why you wanted to image multiple instances simultaneously. Was it, I don't quite remember if in one pot reaction, if you could run many instances of the same algorithm, say parity, or were you able to mix algorithms as well?
Damien tried this once and it didn't work, and we're not sure why. And we thought maybe, I don't know, he just made a mistake mixing or something. So let's try it again. I tried it once and it didn't work, and we're not sure why. And this was like after I started my faculty position. It was just back to Caltech for a week to do this one last experiment. So I don't see any reason that it couldn't be done where you put many different seeds in the same pot and basically have the same tiles doing the same computation but on different inputs. We both thought, yes, of course that should work. So I don't know why it didn't. I think it might have just been something that if that had been like a central goal of the project, we would have like dove in to debug it, but it's like, it didn't work. And having each input be in its own separate tube was enough, and then mix them later to image was enough to like tell that it worked. But I don't see any reason why you couldn't have different seeds with different inputs, but the tiles doing the same circuit. It's just, we don't know why. It was like, and I actually don't even remember right now what went wrong. Like, I think we just might have seen no growth. Like when we imaged it, we just saw a bunch of seeds with nothing attached or something like that. So it's like, we don't know why that didn't work. So we didn't debug it at all. So it should, but.
Right. So in particular, when the tiles, you're generally always in the regime where the tile concentration greatly, or is much greater than the seeds. And so one should never outcompete the other in sense of starvation. And I guess that would be, if I'm thinking about this right, that would be true even for different algorithms that were all deterministic. And then I think maybe you just get into trouble when one algorithm is doing a deterministic choice of a particular probability of a tile, but another one wants a different choice.
It could be.
Yeah.
Well, and speaking of randomness, there's another thing Damien tried that I think I didn't, which was to randomly choose right. So rather than like take certain inputs we want and mix them together, he tried something where he's just like kind of the adapter strands encode the input. He just put them all in and just said, okay, each seed is going to randomly pick an input. And that also did, again, it should have worked. Why not? But it just didn't. Nothing grew, and we don't know why. I did actually forget one detail about the first one, though. Okay, I sort of, there's one difference, which is it's not a one pot reaction. You want to mix together many inputs. It's not a one pot reaction. Because right now what we do is we mix everything, heat it up to 90, everything separates, and then cool it down. But we're only using one origami for the different inputs. The way we program an input is we put in the adapters representing that input and not the other adapters. So if you tried to put several sets of adapters, it would do the thing Damien tried, which is it'll have to randomly pick the adapter. So the thing I tried was prepare different origamis representing different inputs in separate pots, then mix them together, but crucially don't heat it up to 90, otherwise they'll melt, and when they come back together, they'll remix. So that's different than anything else we did, and something in there is what went wrong, that, you know, we weren't following the same protocol. We weren't heating to 90, slowly cooling to 50. I was just forming those things and then putting them into something that was already at 50, and yeah, my intuition told me it should have worked, but the most important scientific lesson I learned from this experiment is my intuition isn't worth squat on any of this stuff. And so, yeah, I don't know why it didn't work, but yeah, that would have been, I sensed it was somewhere between 1 week to 2 years of debugging to fix that. So we didn't chase it down because we had something sort of good enough by keeping everything in separate pots.
Got it. Any other questions from the audience?
How long are the strands you're using?
They're 42 bases. So the domain lengths for each of the 4 domains is 2 of them are 10 and 2 of them are 11. So, which, you know, and then we like designed the DNA sequences so that the length 10 ones were a little stronger than your average length 10, and the length 11 were a little weaker than your average length 10. So they all had about the same binding strength, with which, you know, we use sequence design software, but probably what it did under the hood was put maybe put an extra GC pair in the length 10 and an extra AT pair in the length 11. But they were all like targeted to be about -9 kcal per mole binding strength to their perfect complement. Whereas if you just randomly generate them, you look at the histogram of binding energies. It's that thing I showed you where it could be as, you know, like -5 to -14. They have this huge variance if you don't design them to be close to the same binding strength.
And the origami, the M13 virus is a few K long. Is that right?
That's about 7,249 bases. And then each staple is about 32 bases. There's some variation in that, but the 32 base strand, what it is, it has, I didn't really show a diagram, but it has a length 16 domain in the middle, and 16 is much longer than 10 or 11. So that's the key thing to folding up the origami. The other two are kind of easy once you get there, because then it's a unimolecular reaction to kind of fold it up. So that sort of reasoning together with experiments, we know the origami forms very stably at a higher temperature than the one that you need to get to before the tiles start growing. So this is something we test. Like if you would cool from 90 to 60 degrees and wait like 20 minutes, the origami forms just fine. You can see it, but tiles aren't growing at all. It's too hot for them to grow. They have to get down to like 51 before they'll start growing. And it's because those middle domains of the staples are a lot longer.
Thanks.
Yeah, so just following up on that point, Dave, if you wanted to scale up from 6 bits to, you know, maybe 12 or 15 bits, do you think the bottleneck we would run into is you'd want a bigger seed, so you'd want something like an origami or a super origami that could fold up, or would the bottleneck be the fact that to get really good sequence design, maybe you have to run the experiment at a higher temperature, but you would start working with longer strands, which would essentially form the tiles to give you a bigger sequence space?
I think we can get a lot out of this length of sequence. Again, there's a limit where you get too many tiles, but they're all length 10 and 11 domains. You're going to run out of domain. I mean, obviously you'll run out of domains when you hit, you know, the limit length. There's only about 1 million domains of length 10. But of course, we know long before that we'll hit the limit where like, well, you're not using every sequence, but like now you've got so many that, you know, some of them are so similar to each other, they're going to have this cross binding that you don't want. So, so clearly you'll hit some limit, but I think you can get quite a bit more than 355. In our case, we have a library of 355 tiles, but we're not mixing all of them. We pick a subset of 100 of them to implement this. So we only ever have 100 at once together. I'll bet you can have like, you can get bigger than that and still get good sequence design. We did actually try for the first 18 months of this project was me trying to get tiles to grow from a seed twice the size, from a 32 helix tubular seed rather than 16 helix. And I never got it working, and I don't know why. It was one of those things where it was like 18 months of work, and we just learned nothing other than the bit that didn't work. And I was so sick of it, I was like, okay, Damien, I know you've grown tiles before. What's the biggest circumference you've ever grown? He said 16. I'm like, okay, can you give me the tiles? And he didn't use a seed, but he had tiles that grew into a tube. Like, can you give me those tiles? I'm going to use those, and I'm going to start from something that works and then go. So we never figured out why that didn't work. I think it was a geometric problem. I think it's because the tiles kind of are constrained such that they want to be in a 12 helix tube. And the bigger you make it, kind of the more strain there. I believe that's one reason why something like that was going on, but really, I don't know. I just know that I never got 32 helix tubes to form well. I knew Damien had gotten 16 helix tubes to form. So I'm like, all right, let's just do that. And then we'll make 16 helix tubes. And yeah, it was enough for 6 bits, but had we used 32 helix, it would have been enough for 14 bits. So
right.
Ultimately, I think doing it in a tube was the main limitation. So we mainly did that because that got us around the problem of some technical problems related to the geometry of these tiles, where if you want to do it not in the topology of a tube, but in a flat ribbon or a flat like rectangle, but to do this cooperative binding, you need to design different kinds of tiles than just the ones we used. And this was Eric Winfrey's advice at the start, like, we know that these kinds of tiles form well. You'd need to design different motifs to make double tiles or something if you want to do it in a flat sheet. And yeah, that could be possible. And maybe you'd figure out how to do that in a month, or maybe that would be a whole like PhD project level thing. So we just went around that problem by using tubes. But I think you could do a lot more information processing if you didn't have that constraint that you got to wrap around and do a tube. You just had this big 2D sheet to explore. So
great. But we will end today's talk, and everyone please unmute and thank Dave for a wonderful talk.